So this is a review video for the exam 3, which will cover the chapters 10 and 11. So you should have been provided the blank version of the study guide. So the study guide with few answers missing, so gaps here, every there and now on your Blackboard shell. So it might be helpful to either download that or print it, and then you can fill in notes as we review this study guide together. That works for some students. Of course, some other students prefer to not do that technique and they prefer to write all their own notes, or some students might prefer just to simply listen. So you do whatever works for you. That's what's important for me, that you get most out of this session. But during this session, we will go through the material that's essential for your exam 3 that relates to the chapters that we have covered. And every question in the exam is either directly from this study guide or questions that you can work out based on the information provided on this study guide. So there's no curveballs there. And we're going to start with the introduction to the cell reproduction. And one of the examples that I used during the lecture videos was sea urchins, I believe. And I highlighted how there's such a remarkable variation of specialized cells present in sea urchin, and a mature sea urchin in particular, and how all these cells are so different, yet they have started as a single cell. And this applies to also, of course, other mature multicellular organisms, such as in this introduction, I've discussed human body and how various different cells within a mature human differ from other cells, but yet still they all originate from this one single cell type. So I think that that's something pretty remarkable and kind of a good introduction to the importance of the cell reproduction, why this is something that we want to know about and why this is something that we want to appreciate it. So in the duration of the chapters 10 and 11, we actually had a look of two different cell division processes and how they are both necessary, but how they also both differ from one another. And the two cell division types that we had a look of were mitosis and meiosis. And we started with the mitosis. In mitosis, we are producing cells for all other cells in the body, with the exception of the sex cells. So sex cells have a different reproduction process than mitosis, and we discussed that on the chapter 11 as a separate concept on its own. But what we see with the mitotic cell division is that this serves many purposes, such as growth. We grow bigger, we grow more diverse, we grow having more structures, we grow having specific structures by our cells going through the mitotic cell division process. Also, certain tissues might need to be repaired. For example, when you get a cut on your skin, the repair process of that involves mitotic cell division. So that's something to bear in mind as well. In addition, of course, we are constantly losing cells in certain parts of our body. I think that, for example, hair is a good example. Skin's outermost layer is a good example. We constantly lose those cells as just part of the normal progress of a life. So while we lose those cells, we need to replace them as needed. So mitotic cell division serves also the tissue maintenance, giving us the repairing those cells that we lose during normal life cycle. What's important to note about the mitotic cell division, that the cells that are result in the process, so the resulting cells, we call them daughter cells. So the daughter cells of the mitotic cell division process are going to be always identical. So they're going to be identical to their parent cells, but also identical to each other. And what's also noteworthy is that since they are identical cells, they contain 100% of the DNA. So all of the DNA of the original cell. So they really are an exact copy of this parent cell that we have here. The resulting two daughter cells are going to be exactly like that. So they're containing the exact same amount of DNA and exact same DNA than the parent cell. And also comparing from one daughter cell to another, they are going to be identical. So that's a good overview of mitotic cell division, mitosis, and we will have a look of that as the first cell division process. After that, we will come back to the cell division process, but we will have a look of the meiotic cell division. Meiotic cell division is very different from mitotic in few ways. And probably the biggest difference is that we only see the meiotic cell division taking place in production of two types of cells in the body. So that's in the production of the sperm cells and also in the production of the egg cells. So the reproductive cells, these cells are known as gametes. So gametes are produced as a result of the process of meiosis. So that's another important thing to note here. Well, what happens in the production of the gametes, and we will go through this step by step, but we start with the original parent cell, and that cell divides into two cells. And these two cells divide again into two cells. So as a result of the meiotic cell division, we will end up with actually 4 daughter cells. And all of these 4 daughter cells will have, and I'm just battling here with the pen, I think that that's better. All of these 4 daughter cells will have unique combination of DNA within them. So, because we are preparing sex cells, each sex cell will contain only 50% of the DNA of a normal cell. And this is important because if you think about it, for our sperm and egg cell, we have one of each, and if both of these would contain 100% of the DNA each, we would end up with way too much DNA in the resulting offspring. That offspring would have 200% of the DNA of a normal cell. And of course, that's not what we are after. So we end up producing meiotic sex cells so that sperm cell can contain 50% of the genetic material and the egg cell can contain 50% of the genetic material. If you add these together in the resulting individual, that individual is going to have 100% of the genetic material. So that's the brief explanation why meiotic cell division actually contains 2 rounds of cell cycle and why all of these resulting daughter cells are going to be unique. They're unique, differing from each other, but also differing from the original parent cell. So we want to have as many different kinds of sex cells, as many different kinds of gametes as possible, because this variation and differences between the gametes is what allows us to end up with so many diverse combinations that we all have when we look at each other. We have unique traits, we all look different from each other. So that's thanks to the meiotic cell division and thanks to the sexual reproduction, where we inherit half of our genetic material from one parent and half from another. So these are the 2 types of cell division that we will be looking on this exam. Before we start talking too much about the cell division, we need to go through certain basic terminology that's important for us to be comfortable with. We want to be certain that we know the key terms, we know the key concepts that we will be using so that we can understand what happens in these cell division processes. Okay, let's have a look of what we see here. So first of all, here we see a cell. And within a cell, we will notice that there is this structure that often stains dark, especially when we use various stains to highlight this structure. And this structure is, of course, going to be our nucleus. So let's add our nucleus to this diagram. And we will do so, provided that the computer will cooperate with me. So we are looking next here within the cell. We see the nucleus. So nucleus is where all the genetic material of the cell is housed. So that's the reason why we are going to be focusing on that within there. Well, another thing that we end up seeing, that during the cell division, the DNA within the nucleus actually condenses into these structures, which we know as chromosomes. So chromosomes are these X or H-shaped structures, if you wish. So they are simply condensed DNA, and there will be multiple chromosomes in each cell. If we look at what a chromosome is made of, we will actually find out that a chromosome is made of something known as chromatin fiber. And this chromatin fiber is really the genetic material. There might be some proteins involved in that. And on this figure, I have illustrated it as this kind of a thin string that kind of passes from one bundle of proteins to another. So that's going to be our chromatin fiber. Like I mentioned earlier on, just when we were just now talking about it, chromatin fiber tends to wrap around bundles of proteins. So there's multiple proteins. They look a little bit like balls packed as one. And These bundles of proteins are known as nucleosomes. So you would see these here in our diagram. I apologize that the drawing is not super accurate. That's because this software really isn't meant for drawing, but we're using it regardless. So the chromatin fiber wraps around nucleosomes, which are bundles of proteins. If we look at the chromatin fiber closer, we will of course notice that chromatin fiber is our DNA. So DNA is this sugar-phosphate molecule made of sugar-phosphate molecules, or made of these molecules that are made of these molecules that contain sugar-phosphate. They form the backbone of the DNA single unit of a DNA, which is a nucleosome. And in addition to that, we are going to have various bases. And these bases are the ones that vary. If we look at the structure of a single nucleotide, so an element of the DNA, we end up seeing, and I'm just scrolling up to make some space for myself, we notice that nucleosomes are made of a sugar and a phosphate group. And these are being repeated time after time when we do end up forming the DNA strand. So they're kind of a structure that keeps repeating and repeating and repeating. And for that reason, since they're always the same, we tend to call this as a sugar-phosphate backbone of a DNA. So you'll see how there I'm making two strands, and each strand contains both the sugars and phosphates. And let's see if we move. Oh, let's not move that. So in addition to that, we have something else within the single nucleosome. We have a varying combination of bases. So these bases vary depending on what we are coding. And it is actually these bases that connect these two strands of sugar-phosphate backbone. So this is what we are seeing actually here on this diagram. These ones here would be the bases. And the two sides, so two backbones, which we looked at and were made of sugar and phosphate. Oh, I didn't mean to change the color of that, pardon me. Now let's jump back. They would be these continuous sides of a DNA strand. So one way to think about the structure of the DNA that many of my students have found quite useful is to think of it as a ladder. So we have these vertical sidebars, and these vertical sidebars would be, in this case, if we draw an analog to a DNA structure, they would be our sugar, made of our sugars and phosphates. So they would be this sugar-phosphate backbone. And in addition to that, we will then have these horizontal beams, which are actually made of two parts, each one connecting on in the middle. So these are our bases. And you remember it was the bases that varied. And then we take this ladder and kind of twist it around itself. And that gives us this appearance of a DNA that we're very used to seeing. That's the kind of what we think when we think of what a DNA strand looks like. So I hope that explains some of the basic terminology. We want to be comfortable with that. And one more thing that I want to highlight here is going to be, and I'm going to just pause, is going to be pardon me. So one more thing that we're going to see is that the section of a DNA that codes for a particular trait, so just a part of a DNA, and this codes either for a specific protein or a specific trait, so this section is known as a gene. So I hope that those make sense and kind of clarify of what we are talking about when we are talking about some of the basic concepts that we see here. Next, I want to mention a few words about these chromosomes. So we go back here where we already saw the chromosome structure, and we will talk a little bit more about this, having a little bit of a closer look at what we need to know about the chromosomes. So chromosomes are made of various different components, and we will pay attention to that as the next thing that we need to see. So what we'll notice is that a chromosome is made of 2 chromatids. These chromatids are, like if you wish to make a comparison, sticks that form the sides of the H or X letter, whichever you prefer to view a chromosome as. So here we would have 1 chromatid, and then let's have a look of a second chromatid. So each chromosome is made of 2 chromatids, and these chromatids are held together by a structure known as centromere. So centromere is the structure that holds the chromatid together. And I'm just gonna try to add that there. So this would be our centromere, holding the 2 chromatids together. And usually the centromere is located roughly in the middle of a chromosome. So those are the structures that make up the chromosome. In addition, I want to highlight 2 more concepts, 2 more terms about chromosome. First of all, each chromosome has particular locuses. This locus is where a certain gene is coded. So we end up finding that the genes are coded on both chromatids and at the same location of the both chromatids. So if here is a locus of a certain gene in one of the chromatids, we would find the same gene coded in the other chromatid at the similar locus. Well, another thing that we discussed during our review of the chapter material was that we could have genes, and genes could have many different alternative forms. So I think the example that we looked at was, for example, how your hairline is at the very front of your head, on your forehead. And we saw that some people had, and I'm going to draw here, just a very simplified diagram of a head. Here's the eyes. And we have a mouth. That's a happy person. And we're going to have another happy person because we're going to compare 2 different kinds of options that we can have. So, example of the hairline was what we were looking. So let's look at an allele that codes for a widow's peak as a hairline. Widow's peak was this sharp kind of a triangular end of how hair goes at the forehead. And that was a dominant gene, so we're going to symbolize it with a capital W for widow's peak. Other people could have a straight hairline. This straight hairline was a recessive character, so it's going to be a lowercase w. It's not as common. So what we find that whenever we have an individual with a dominant gene, we tend to see that trait. If we have an allele for a recessive, so lower case letter, that's not as common characteristic, we see those less often. So a single locus would hold these alternative forms of genes for a certain trait in each chromatid. And the important thing to do here is to note that these alleles don't have to be same within one chromosome. We can have two different alleles within one chromosome, so different allele on each chromatid. And I kind of went a little bit ahead of the game already, so let's just catch up as a review and look what we know about alleles. So allele codes for a particular gene, and there's actually two alleles in each chromosome. As we saw here with this, I'm going to add this. So we saw that in this case we had a dominant and recessive allele for the hairline type. And we inherit one from each of the parents. So we have different forms of that. And we saw that if we had a dominant allele, which was this capital letter, so in our case of an example, it was the widow's peak. It doesn't matter if you inherit one from one parent or if you inherit one from both parents. So as long as you have one dominant allele in the offspring, we will always see that characteristic as the visible trait. We could also inherit recessive alleles. Recessive alleles are lowercase letters, and they are less common in nature, and we see that only if we inherit a recessive allele from both parents. So for this reason, it's not that common to see recessive traits in the offspring or in the nature. So having a straight hairline, as in our example here, that is going to be less common, whereas having a widow's peak is going to be more common. And that's because the widow's peak was a dominant allele. So I would like you to be somewhat familiar with the concept of dominant and recessive allele. Other examples that we could consider is, for example, how your earlobe is attached. Is there a clear separated earlobe, or is it smoothly attached to the rest of your head? So that would be one characteristic. There's many other characteristics, and some characteristics get even more complex than having just two alleles. Other characteristics that I could think of would be, for example, eye color, hair color, and so on and so on. So we're going to keep it simple and look at this widow's peak or straight hairline as an example, because that stays nice and simple with just two alleles that play a role. I also want to mention, just kind of to clarify some of the language that you can run into, that when we talk about genotype, Genotype is the genetics of an individual. So what we are doing when we're looking at dominant and recessive alleles, like we've been discussing in these examples of that, we are talking about the genotype of an individual. So we're looking at the genetic coding that a certain trait, for example, can have. Phenotype instead is going to be what we actually end up seeing. So the visible trait or visible characteristic, as in this case, it was going to be the type of hairline that we see. So the type of hairline that we see is going to be the phenotype, and genotype is going to be the coding, either as capital W, or lowercase w. So there's a lot of material that we have covered here, I know, but I do want to ensure you that it is an open book exam. You can keep all of these notes with you, so that should eliminate some of the stress and concern. I'm really not trying to catch you out on the test. It's all concepts that we have discussed about. So before we get too much further ahead, there's also two more terms that relate to everything that we have discussed so far. And they relate to the fact whether we see an individual having alleles that are the same. So those two alleles that we inherited to our chromosome, if they are the same, either capital letter and capital letter, or alternatively lowercase letter and lowercase letter, we talk about homozygous alleles. So these alleles are the same. There's many different ways to think about that, and sometimes students struggle with it. But if you think about different kinds of couples that we can have, if the couple is the same sex, we can talk about a homosexual couple, for example. So homozygous means that they are the same. Let's have a look of heterozygous combination of alleles. In a heterozygous combination, we have different alleles on each of the chromatids. So the same chromosome contains two different alleles. So if you think of our relationships where we have hetero relationship, we have two different sex involved in there. That might help you to remember it. I don't know. That's my attempt to simplify it. Let's look forward what we need to know. So these have been some of the basic concepts that I would like you to be comfortable with. And one of the main topics on this chapter or these chapters was the cell cycle. And we're going to be talking about the cell cycle as the next thing a little bit. So let's have a look what a cell cycle means. Why do we need to know about a cell cycle? So cell cycle is a general term that we use to describe all events that take place during cell's life. So cell starts its life at one point, and then various different events proceed throughout its lifetime until the cell stops existing. All of these events are part of the cell cycle. And in addition to this kind of yellow part that I've drawn here, also this turquoise part that I've drawn is part of the cell cycle. And I'll explain in a moment why I drew them with different colors. But all parts of this circle that we see here are parts of the cell cycle. So what we end up noticing is that these events during cell's lifetime always proceed in an ordered manner. So we go from one step of an event to the next step. So it's always proceeding in a very particular manner. And let's have a look at a few of the steps that we see during cell's life cycle. So we end up seeing that there are a number of steps during what we call interphase. Interphase is the part of the cell's lifetime during which cell grows in size, and in addition, the cell also increases the number of cell organelles within it. So we end up seeing that during interphase, cell goes through increase in its size, but also increasing the number of cell organelles within the cell. And this interphase serves a purpose that the cell is growing bigger so that it can divide at the end of its cell cycle. At the end of its lifetime, the cell will divide into two. So that's the general gist of what interphase means. So interphase is this period of time during which cell grows in size and cell also duplicates the cell organelles within it. There are different stages within the interphase, and we're going to have a look at those. And although they're not listed in this physical review guide, they are something that could be included in your exam as a question. So we will make a note on this sheet together about the important things that you need to know about various steps within the interphases. And we're going to start with G1 interphase. And as you'll notice here, the G1 interphase is the first stage of cell's life. And let's have a look at what happens during G1 interphase. During this time, the cell grows and it grows both in size but also the number of cell organelles gets actually doubled. So we saw that these cell organelles, when we reviewed the parts of the cell, these included, for example, mitochondria, these included Golgi apparatus, these included endoplasmic reticulum, these included ribosomes. There's a lot of other parts that are included in that. So all of those cell organelle structures inside the cell, they duplicate. So we want to have larger cell and we want to have more cell organelles inside it. Well, the next step that we are going to look at is gonna be our S interphase. So you'll notice in our diagram, S interphase is gonna be the following part of this cell's life cycle. During S interphase, the cell's DNA is replicated. So we end up making sure that the cell has double the DNA within it at the end of S interphase. And this is again in preparation that the cell might later on end up dividing. So we want to make sure that there's twice the DNA so that each resulting cell, daughter cell, will have a full set of DNA. And the final step of the interphases was the G2 interphase. And let's have a look what happens during the G2 interphase. And you'll notice that it's the final step of the interphases, and it's not quite as long of a stage. Oops. So I was meaning to put number 2 there. So during G2 interphase, what happens is that the cell grows more in size, and now it is going to be ready to divide into 2. So that's our G2 interphase. Okay, so those are the stages that cell spends most of its cell cycle. So the biggest part of cell's lifetime is spent in this interphase. So that's the majority of cells' existence spent on these stages that prepare it to divide into 2. And for the exam, I would know what happens within each G1, S, and G2 interphase. You have a nice note there that we wrote together while we reviewed this, so you should be well set for that. Well, we have also one more part of the cell cycle to look at. And this part you notice that I highlighted in kind of a turquoise color. So this is going to be the actual mitosis. And what happens during this part is that the cell actually divides properly. So this is the actual cell division process. And what you'll notice that at the end of this, we end up with 2 cells. So at the end of a cell cycle, we have 2 cells that result from it. And this is all thanks to the mitosis. So mitosis is not a major part of cell's existence, but it's the part that results in 2 final cells. And we're going to spend a little bit of time looking at mitosis, and that tells you that even though in terms of the amount of time it's not quite as important, it is an important in terms of events that take place. I do also want to introduce one more term for you, and this term is going to be cytokinesis. Cytokinesis refers to the division of the cytoplasm and cell organelles. It does not include the division of the DNA. So during cytokinesis, the cytoplasm of the cell and all the organelles, with the exception of the DNA, are divided into 2. Well, this is what we know about the cell cycle and the different phases of that. If you think about it, it would be easy for us to have errors in this process. This seems like there's a lot going on and a lot of chances where things might not go perfectly. And we don't end up seeing that many errors in it in real life. If you think about the errors in the control of cell cycle are usually cancers. So the cell cycle regulation has failed when we have cancerous growth of a tissue. The tissue grows in uncontrolled manner. Well, the reason why we don't end up seeing that many errors or mistakes in it is because of these cell checkpoints, cell cycle checkpoints. So they do a fantastic job of controlling this process and making sure that if there is an error somewhere, that error must be either fixed or then the cell cannot continue its cell cycle. And we're going to look at different kinds of checkpoints that we have. Well, the G1 checkpoint is the first checkpoint that we have. And during G1's checkpoint, you might remember that this is the most important checkpoint out of these all ones, because once the cell has passed this G1 checkpoint successfully, it is now committed to divide. It is going to divide after this checkpoint. During this checkpoint, we are checking the DNA for any errors. That is the DNA correctly copied? Is the two sets of DNA that we have, are they correct? There should not be any errors within there. If there is an error, we have two options. The cell might either fix the error, and after it has been fixed, the cell is then able to proceed to the next step of the cell cycle. If this DNA error cannot be fixed, the cell cycle stops. Cell will not progress any further in its cycle. Okay, let's have a look of the next checkpoint. The next checkpoint is known as G2 checkpoint. And during this checkpoint, we are checking, the cell is checking, cell is verifying that it is ready for the DNA replication. Is the DNA correct? That there is no errors in that. DNA can be replicated. We can make 2 copies of that. And the final checkpoint that we have is gonna be the mitotic checkpoint. During mitotic checkpoint, we are actually checking that all of the chromosomes within the cell have attached to spindle. And you might be wondering, what is this spindle? We'll talk all about it in a few moments. But if all of the chromosomes have not attached to the spindle, by the time the cell reaches the mitotic checkpoint, the mitosis will not proceed ahead. There's an error. The cell has not accomplished the goal that we have set for it, so the cell will not proceed any further. I hope that makes sense. There's a little so that's all that we need to know about the cell cycle. There's a little section of a couple of other concepts typically at the end of our discussion of mitosis and meiosis that I also included, and I would ask you to be just aware of these. There might be some questions, but these questions would not be in depth about this. So we can control the cell cycle in various ways. So we are constantly adjusting, changing the cell cycle depending on the needs of the organism. And let's start by looking at stimulation of the cell cycle. So stimulation will make the cell cycle proceed. And several things that can stimulate the cell cycle are going to be, for example, various hormones, different growth factors. If you think what a growth factor would do, well, it makes the cell grow. So it makes sense that growth factors would be increasing the progress or speeding up or promoting the progress of a cell cycle. Well, what else can we have happening? We can have, for example, inhibition of the cell cycle. So when cell cycle is inhibited, we are slowing down the progress of the cell division. And let's look at an example of that. And I think that the example that I gave you in the lecture video was that if we thought about cells within a dish, let's say a petri dish, here, within a glass dish, we have some kind of material that's ideal. Let's say that it's an agar of some kind of a food for the cells to grow in. That's at the bottom of the dish. And the cells are happily growing there. So cells are going through the cell cycle, dividing and making more cells and more cells and more cells until they reach the edge of this petri dish. And when they reach the edge of this petri dish, that triggers cell cycle inhibition. So the physical contact with the edge of the petri dish tells our cells that now there's enough of you, now we've reached the boundary for you, we are going to inhibit your division. So that would be inhibiting the cell division. And the final concept that I discussed with you guys, which I think is very interesting and very fascinating, and there's a little bit more to that story, but we're going to stick to the level that we discussed, was the fact that we allow cells to be copied only so many times. The DNA within the cell can be copied only so many times. So if you think about it, with each time that a cell replicates, so I'm only drawing one of the cell, it's going to produce 2 cells with each division. But each time that we're making a new cell, each time the DNA within that cell must have been also copied. And what happens if you take a photocopy of a photocopy? Well, the resulting photocopy is not going to be of an as great quality. We've all seen that, especially if you're an old school student like I am, when photocopies were largely used by the teachers for materials, the quality of the result will be lesser. Well, the same is for the copying of the DNA. With each copying process, there is a chance that we make mistakes. Usually these mistakes are very small, just a couple of bases here and there. So couple of these sticks that we saw here. It might be just one that's not correct. And most of the time what happens is that they don't actually cause problems for us. Most of the time these errors just go by. They're not in a part of the DNA that codes for a protein or codes for a trait of some kind. So they just get past that and it's not a big deal. In addition, another thing that can happen is that we have spontaneously arising mutations. So there's just random changes in the DNA. This just happens spontaneously. We can have also mutations due to environmental factors. So, example, if you are big on sunbathing, your DNA in these cells that are at the surface of the skin, they might be exposed to ultraviolet radiation, and they might end up having mutation. That might end up causing mutations on the DNA of these cells. So all of these little errors, they end up adding up because every time we copy that error to the next copy. So we're not going to notice that there's an error until they keep accumulating and accumulating on each other. But nature has a clever way of giving only so many copies to be made of DNAs. And here I've drawn a kind of a triangle, and that, or drawn a tube, not a triangle, drawn a line that shows a DNA strand stretched out. And close to the end of that DNA strand, there's going to be a section that codes a message that do not copy this DNA strand anymore. But this will not stop the DNA copying as long as we have some more DNA after that. So this will not play a role here where we have extra DNA strand at the ends before this command, do not copy any further. But what happens every time that we copy a DNA, we lose a little bit from the edges of it, and we approach closer to this stop command, where we're being told no more copies to be made. And eventually, these stop commands are the very last pieces of this DNA. Now that they are the very last pieces of this DNA strand, well, now there is not going to be any more copies made of this DNA strand. This is the code or the signal that we give to the copying process of the DNA. We've made enough copies of this. It's like we've made enough photocopies of photocopies of photocopies. Now we are going to start to get concerned that the quality is no longer ideal. So nope, you have to find another cell where we find DNA that has not been copied that many times, so that we can be sure that the quality of the DNA is great. So that was the example that I had about the limited number of cell divisions that can take place before the DNA is no longer available to be copied. The other concept that we saw at the end of when we talked about the process of meiosis was a karyotype. And what a karyotype was, let's have a look at that a little bit. So karyotype was a name for this figure, this visualization that shows all of the chromosomes of an individual. So there's 23 chromosomes that we all have. And these chromosomes in a karyotype are organized based on their size. And we organize them from larger to smaller, and we end up noticing that there's going to be 23 of those in all healthy individuals. That's 15 and so on until we get to that 23. And we can look at this karyotype because this karyotype will tell us a lot about the individual. And usually this karyotype would be taken from an unborn child. So we're wanting to see, is everything okay with this unborn child? Well, we want to look that is there a right number of chromosomes. For example, if some of the chromosomes are missing, that usually means that there's going to be a genetical disorder in this child. Sometimes these can be rather serious disorders, so we want to check for that. Just so we know at the early stages of pregnancy, is everything the way it should be? We can be also looking at the size and shape of a chromosome. Is the size a normal size? Is it what we expect it to be? Is the shape of the chromosome the same in both chromatids? Is there something abnormal there? Is everything the way it should be? If there are abnormalities, we might end up doing further testing. And like I said, that was a signal that there is probably likely to be some kind of a genetic abnormality in that individual that's developing. We can also see, of course, the sex of an individual from the karyotype. And what we saw, that when we had XY, that was male. and XX was female. So it's the chromosome 23 that tells us the sex of an individual. So that's often an important one to check if we want to define before the birth genetically the sex of an individual. So those were the kind of side stories, if you wish, about that I added to our chapters of mitosis and meiosis, because I think it's always good to expand a little bit on what we have been discussing. But now, the remainder of this review session, we're going to be looking at the process of mitosis and meiosis. And that's actually not going to take a huge amount of time. So let's go through and see what we see happening in each of these. And let's start by going through the stages of mitosis. So mitosis was the production of all cells in the body, with the exception of the gametes, with the exception of the sperm and egg cell. And mitosis was the cell division where we ended up with daughter cells that were exactly alike. So they were identical to their parent cells and also to each other, the resulting cells. And the stages of mitosis are going to be prophase, metaphase, anaphase, and teleophase. So 4 stages for us to know regarding the mitosis. And what we are going to do as part of this process, we're going to draw the cell division process at each stage that we're looking at for the mitosis. So let's start with the prophase. During prophase of the mitotic cell division, here we have the cell, but we notice that we notice that the nucleus of a cell, which we would normally see in an initial cell, so this is not prophase that I'm drawing here. This is a normal cell. This nucleus would be present, but it disappears by the time we get to the prophase. What we are going to see within the prophase stage is the centrosomes appearing instead. And you remember these were from our review of the parts of the cell organelles. This was this pair of kind of a tubular parts, 2 tubes that appeared. In addition, from these centrosomes, we have a bunch of fibers coming out that are known as spindle. So spindle also appears during the prophase. And this was what I referred earlier on, that we would talk about it later on. We will see that there's gonna be DNA in there. If we do see chromosomes, the chromosomes are all over the place. They're not in any organized manner in any particular part of the cell during the prophase. Let's move on to the metaphase and see what happens there. So now let's start by drawing the cell again. And within this cell, we will see again I'm trying to show the parts there. Good. We're still seeing our centrosomes. So these pear-like structures. And we are going to see our spindle branching out from the centrosomes. What's noteworthy that happens during metaphase is that now all of these chromosomes that we saw floating freely wherever during the prophase. Now they all line up. They go on an organized manner, on a straight line on the cell, and this line that they form that we're seeing is known as this location is known as Equatorial Plane. So if you think of the equator of the Earth, so here we have the Earth, we have our North Pole, and we have our South Pole. So we have an equator going right on the middle of the Earth. And that's usually the part that's closest to the sun. That's the warmest part of the Earth, and so on. Arizona is fairly close to the equator, so that's why we get so much sunshine. That's why it's so warm here, and so on. Where I was raised was right close to the North Pole, so we certainly didn't get a lot of sun. But I'm not going to teach you about geography. That's not the topic of this course. But what I wanted to highlight was this equator. So the same term, something that goes right in the middle of a planet or right in the middle of the cell. That would be our equatorial plane, which we saw that all of the chromosomes aligned on the equatorial plane of this cell that is going through the mitosis. Let's move on to the anaphase. What happens during the anaphase? What kind of things can we see taking place? Well, now here we have this cell. Notice that it's a little bit of a different shape than before. And I'm going to just redraw it because I don't think that that was very beautiful drawing that I did there. Maybe this is a little bit better. This cell is now kind of turning more into a sausage shape. It's a little bit spreading out, but we still have our good old friends, centrosomes, and they're at the different ends of the cell, so different poles of the cell. And in addition, we're going to see our good old friend. Oh, I didn't mean to change the color of that. I meant to change the color here. We see our good old friend, the spindle, as well. There we go. That's our anaphase. And what you saw on this equatorial plane in our previous stage, the metaphase, the chromosomes were attached so that they were attached on both spindles. But now these spindles start pulling apart. And as they pull apart, each chromosome gets split into two. So we have only the chromatids left for each chromosome. So we're pulling the chromosomes apart and we're left with the chromatids, so 1/2 of a chromosome. And these halves are pulled to the different ends, different poles of the cell. Another thing that we're seeing during the anaphase is this. When I was drawing the cell, you notice that I made this kind of a dip in here, kind of a depression. This is where the cell will eventually be pinched into two. So this is the early formation of where the cell will be pinched into two. So this part is known as cleavage. So that's starting to appear. We're seeing how the cell will be pinched into two. And finally, we get to telophase. During telophase, now we see two separate cells that have resulted from this process. And in addition, let me draw those again so that they stick within the boxes. In addition, we will see that the nucleus becomes visible again. So I'm just circling so you know what I'm drawing here. This is going to be the nucleus, so the nuclear envelope and the nucleus reappears. And both of these cells will have now DNA matter. So that's the process of our mitosis. And I'm just jumping right to the front to look. I used green color for the mitosis so that I'm being consistent with the color coding from one area to another. So this is the process of mitosis. Mitosis included stages prophase, during which the nucleus and nuclear envelope disappear, centrosomes appeared, and spindle appeared. Notice how the chromosomes are floating all over the place. Then during metaphase, the chromosomes attach to the spindle and line up on the equatorial plane. During anaphase of mitosis, the chromosomes split into chromatids, and they are pulled towards the different poles of the cell, and a cleavage starts to appear. So this initial pinching into two. And finally, during telophase, we now have two separate cells, and the nucleus, nuclear envelope, reappear on each cell. And what we have done here, we have produced cells that are identical in their DNA content. In addition, they both have 100% of the DNA of that cell, that initial cell. So both daughter cells have 100% of the DNA. And of course, there's going to be two cells, two daughter cells that result. That was the process of mitosis. And we remember that mitosis was production process that we used for all of the other cells in the body, with the exception of the sex cells. So I hope that that clarified the process of mitosis. And just to remind ourselves, we're seeing all of these things that we talked about earlier on. Mitosis produces all of the cells in the body with the exception of the sex cells. The function was to allow this body to grow, repair any tissues that get damaged, and also maintain any tissues that we might lose just as a part of a normal life. They contain 100% of the DNA in the offspring, so in the daughter cells. And these cells are, in terms of their genetics, identical, both the resulting daughter cells and with the parent cell as well. Well, that completes our review of mitosis. Next, we are going to look at meiosis. So let's jump right into there and have a look what happens in meiosis. You'll notice that my discussion of the meiosis is going to be a little longer. So there's a little bit more going on in the meiosis. And before we're able to look at meiosis in a greater detail, I want to highlight two important concepts that we only see during the meiosis, not in mitosis. And the first one is known as synapsis. What happens in synapsis? We have two chromosomes, and these chromosomes line up so that they're next to each other. So I'm drawing two chromosomes of different colors for a reason that will become obvious in a moment. But these two chromosomes end up hanging out next to each other. They pair up next to each other. That's an important event that happens during the meiosis, and that event is known as synapsis. During synapsis, these chromosomes pair up next to each other. Another thing that we end up seeing happening during meiosis, and only during meiosis, is a process called crossing over. And let's have a look what happens in crossing over. And to explain what happens in crossing over, I'm going to divide this box into two. We're first going to look at what happens initially and what's the end result. So, in crossing over, these chromosomes of ours are going to overlap with each other. So parts of them are lying on top of each other. So that's what happens initially. And during these parts of the chromosomes lying on top of each other, we end up changing little pieces of them. So you'll notice how this initially red chromosome received a little piece of the blue or turquoise chromosome. And this initially turquoise chromosome, now it has received a little bit of the red chromosome. So this is how the chromosomes get mixed with each other during the crossing over. So little pieces of these two chromosomes get exchanged with each other. That process is known as crossing over. So you might wonder, why do we have these two things happening then during meiosis? That just makes things very difficult and complicated. Well, it's not to make your life miserable. It's to introduce a genetic variation to the cells, the daughter cells that will result. We want to make sure that there's a maximum amount of various different combinations that we can have in the resulting daughter cells. So that's why meiosis has these two extra processes taking place, in addition to all other things that take place during the meiosis. But you want to know synapsis as chromosomes pairing up next to each other, and crossing over being this process where parts of the chromosomes are exchanged, and as a result, we are left with these chromosomes that have mixed up parts between each other. And in reality, the crossing over doesn't just happen in one spot at one time. There's multiple parts of these chromosomes that overlap with each other, so we see a lot more mixing up happening. But for the sake of keeping things and diagrams simple, we typically, when we're teaching this, just show one side happening with the crossing over. I hope that helped to clarify the concepts of synapsis and crossing over. And now we are ready to move on to look at the stages of our meiosis. And while we're revising the stages of our meiosis, I want to highlight that we have the exact same steps that we had earlier. However, now we have number 1 at the end of that. So we have prophase 1, metaphase 1, anaphase 1, and finally telophase 1. So they're the exact same steps in terms of the names that we saw earlier on. But what happens in these is a little bit different in terms of appearance. But after that, we have these repeated again, but these are now prophase 2, metaphase 2, anaphase 2, and of course finally our telophase 2. So this tells us that during meiosis, cell goes through the second cell division. So we go through 2 cycles of cell division in meiosis. That's supposed to be N. So second cell division is what happens. So you can figure out that it's going to be a little bit different what we are left with at the end of the meiosis. But let's have a look of each stage of our meiosis. And we're going to start with the prophase. And here we have our initial cell. And again, I'm going to draw one of the very early cell that we see before we go through the meiosis as well, just so I can highlight what happens here. You've noticed that in the initial cell, before we even get to the meiosis, we still had our nucleus there. But once we get to the prophase 1, the nucleus will disappear. That's no longer visible. But what shows up is gonna be our centrosomes. So you remember this pear-like structure. And now let's go back here. I'm gonna circle the centrosomes. So if you are into color coding, that should hopefully make sense. And in addition, the spindle appeared. I hope that those make sense from the past, what we've already discussed. In addition to that, during prophase 1 of our meiosis, something else happens. These chromosomes that are within the cell, they form synapses. And there's also some crossing over happening. So little bits of DNA get exchanged between the chromosomes. And remember, this is going to be 2 chromosomes of the same. So, oh, I didn't mean to take that away. So we're going to be seeing that we're able to exchange these pieces between these 2. So there is both synapses and crossing over happening within the cell during prophase 1. And the chromosomes are floating wherever they happen to be. For the sake of clarity for the rest of these drawings, I'm not going to draw the result of crossing over for these chromosome pairs because it would just make it too difficult for me to draw. I have limitations. I'm only a guy. So let's move on and have a look what happens during metaphase 1. So that was initially prophase 1. Now we're looking at metaphase 1. So here we have our cell. And we're going to draw again our centrosomes. You remember them from the past? And we're also going to draw our spindle. So that's our metaphase 1. During metaphase 1, the chromosomes line up on an equatorial plane just as before. But what's interesting is that they line up in pairs. I'm just going to redraw that. So now, during meiosis, it's pairs of chromosomes that line up on this equatorial plane, which we saw earlier on. So notice how it's pairs, and it's again this equatorial plane. During anaphase 1, we again have this cell. Oh, I'm going to redraw it. We again have this cell here. And we're gonna redraw our centrosomes and our spindle. I'm not gonna scroll up this time. You just know that it's a spindle. I try to scroll up so you can see the color coding every time, but this time I'm just cutting corners. Now, these chromosome pairs are pulled apart to different poles of the cell. So we're not breaking up a single chromosome into chromatids like we did with mitosis, doing meiosis and first round of cell division of meiosis. We're actually separating the chromosome pairs. And as a result, we end up with one chromosome on each, or we end up with chromosomes on each ends. You also notice that the cleavage starts to form, so this kind of pinching off the cell wall when we're getting ready to produce two cells. We saw that before too. And finally, we reach the telophase 1 of the meiosis. And now we're going to have two cells that have resulted from this process. And the nucleus on these two cells is also gonna reappear. So we have now completed the first cell division of our meiotic cell cycle. So that's the end of that as the telophase 1. Now we move on to the second cell cycle. And we're going to go to the prophase 2. During prophase 2, now I'm drawing two cells since we have two cells going on. Maybe I should draw them more centrally. Let's do it here. Perfect. The nucleus disappears. Centrosomes appear, and the spindle appear. And what you'll notice that this second division of the meiotic cell cycle is exactly the same as mitotic cell cycle. The only difference here is that the genetic material hasn't been replicated between these two cell cycles. So the genetic material has stayed the same. We did not double it before going to this second division. I'm going to write that there just as a note for us. And this gives us a clue that we're probably going to end up with half of the DNA as a result of this, rather than the full amount of the DNA, right? So we also end up seeing that we have the chromosomes floating freely wherever they wish within these prophase 2 cells. So they're just wherever they happen to be. Then we move on to the metaphase 2. Again, we're going to draw the cells. We're gonna see our centrosomes. I'm trying to do the color coding here. We're seeing also our spindle. And you don't have to draw the two sets of the cells if that doesn't work for you. I think it's useful because we'll notice something important at the end of this all if you draw the both. But as long as you know that the second cell cycle of meiosis is similar to the mitotic cell cycle, with the only exception that the amount of genetic material hasn't been duplicated before it starts. So now, during the metaphase 2, the chromosomes line up on an equatorial plane. Here we go. I'm just going to go and circle the nucleus here because I kind of feel that I didn't do that and that bothers me. So you remember that this was the nucleus. And then we move on from metaphase 2 to anaphase 2. So notice on the, just as a note, on the metaphase 2, the chromosomes have lined up to the equatorial plane. We've seen this before, but just because there's only 2 chromosomes, it might be harder to see it. But notice that they're on an equatorial plane. Now we get to the anaphase 2. Well, that didn't turn out pretty. Remember, I would never judge your work on your artistic abilities. I just draw things out because it helps me. It might help some other students to know what's going on. But if that doesn't help you, you can just visualize it in your head or make notes in writing, whatever works for you. I think that this is a concept that's kind of hard to imagine, so at least drawing it out helps a lot of students. So that's why I'm doing it visually. So let's see what happens during anaphase 2. Just let me add these couple of last structures. Perfect. And now, during anaphase 2, these chromosomes that we saw earlier on during metaphase 2 they get split into two. No, I didn't mean to do that. These chromosomes get split into two into chromatids. So they're pulled apart. So during Anaphase 2, we're seeing the chromosomes split into two, and each chromosome then makes a chromatid. And they travel to the different poles of the cell, towards the different poles of the cell. In addition, we're starting to see this cleavage again, the formation of where the cells will pinch off into two. So that's starting to become visible as well during Anaphase 2, especially towards the end of the Anaphase 2. And the final stage that we have to go through is going to be Telophase 2. And as a result of this meiotic cell division, we're now left with 4 cells. So notice that. That's something that I want to highlight here. So 4 cells result from this. And during Telophase 2, the nucleus reappear on each one. And we have completed the meiotic cell division. Let's have a look of a little bit, what do we have as a result of all this? So first of all, we have 4 cells. So meiosis results in 4 daughter cells. In addition, all of these 4 cells, each one of them has a unique combination of DNA. Partially because of this crossing over in synapses, partially because of these 2 cycles of cell division. And finally, the thing to notice here is that because we have 2 cycles of cell divisions, each of these cells will now have only 50% of the DNA in them. So we have halved the amount of the DNA because there was no DNA duplication before the second round of cell division. Now we're only going to have 50% of the DNA in each of these resulting daughter cells. So that's the process of our meiosis. And let's go to the very start just to remind what we noticed earlier on. So meiosis was the production of the sex cells, so sperm and egg cells. These are known as our gametes. And during this process, we actually go through 2 cycles of cell division, and these 2 cycles of cell division, without duplication of the DNA in between them, ends up that we end up with 4 cells, each of which contains only 50% of the DNA. And all of these cells, daughter cells, are unique from each other and of course from the parent cell as well. So this is how we introduce the variation to the sex cells, and we have 50% of the DNA in each of the sex cells in a sperm and in an egg cell. When they fuse together, we get 100% of the DNA in the offspring, in the child, but 50% is from a father, 50% from the mother. I hope that makes sense. So now we have reviewed I'm going to add that to the meiosis, that it's the production of the sex cells. So sperm and egg cells. Now that we have reviewed these two, I think it's a good time for us to compare these two as well. So let's compare mitosis and meiosis a little bit. So let's kind of summarize what we've seen so far. We're looking at mitosis, and we're comparing that, of course, with the meiosis. Let's see what we can find out. Well, let's have a look first of all. What type of cells are we producing in each of these? Well, you remember that mitosis produced all cells in the body, with the exception of the sex cells. So mitosis really produces every other cell but the gametes in the body. And you remember that our gametes were our sperm and egg cells. Well, how many cycles of cell division do we go through in each? Let's have a look of that. I think that that's an interesting thing to look at. And we remember in mitosis, we're of course going to do only one cycle of cell division, whereas in meiosis, we had 2 cell cycles. And the big thing to notice here was no DNA replication. In between of these cycles. So that's an important thing to notice regarding the meiosis. There is no DNA replication between these 2 cycles. Now we're ready to look at the number of cells that result. So the daughter cells, how many daughter cells are we ending up with in each? Well, of course, in mitosis, there's going to be 2 cells because it's one cycle of cell division. Whereas in our meiosis, because there's 2 cycles, we end up with 4 cells in total. How about the amount of the genetic material? How much genetic material do we have in each? Well, in the mitosis, we had 100% of the genetic material in the resulting daughter cells, whereas in the meiosis, the resulting daughter cells had 50% of the DNA in them. And here comes some terms that we covered in the lecture videos. You remember 100% of the DNA could be sometimes abbreviated as 2N. So that's the full amount of DNA in the cell. The term that we used to describe that was that these cells were diploid. Dip for 2N. So they have 100% of the DNA in them. Whereas on the cells that result from meiosis, we only have an N per each cell. So you don't need to necessarily put the number 1 if you know the rules of the math. And this is known as haploid. So half of the genetic material in the resulting daughter cells. This would be a good concept to be familiar with and comfortable with as we move on to take the exam 3. So know that all of those things mean the same. 2N is same as 100%, which is same as diploid cell content. And 1N is the same as 50%, which is the same as haploid cell content. And finally, we already saw this, but I want to emphasize how similar are the resulting daughter cells. Well, in mitosis, these resulting daughter cells are all going to be identical, whereas in meiosis, the resulting daughter cells, they're all going to be unique that we produce. So I think that that's a really nice comparison of these two cell division processes. There is one more concept that I want to introduce here, and I'm not sure if we discussed that as a part of the lecture material, but it is a question that might come up in the exam. So you remembered when an animal cell, and we've been looking at animal cells in all of these examples so far. When an animal cell was about to divide into two, we had this cleavage forming. So the cell membrane simply started to pinch down, and eventually it just pinched these two cells apart. So we ended up with two separate cells. So the cell membrane simply moves inwards towards each other and eventually pinches it into two separate cells. So this is the case in the animal cells, where the outermost layer of the cell is this soft cell membrane, and I'm adding the nucleoid there. How does this work in plant cells, you might ask? Well, we remember in plant cells we had rigid cell walls, so that's not going to work that it could pinch in. What happens in plant cells is that we simply start building this cell wall little by little by little, and it keeps growing up until it reaches in the middle, and at that stage we are left with two separate cells. So rather than the cell wall really kind of bending and pinching into separate cells, in plant cells it is gonna be this process of slowly building the wall that separates the two cells from each other. So that's how plant cells end up separating into two resulting cells. So that's just a concept that I think is important for you to know as well. I do think that there might be a question about that in the exam. I'm not making any promises though, so that's one more aspect to know. But I think that at large finishes off the revision of the lecture material that we had on chapters 10 and 11 in preparation for the exam 3. That's the study guide and review of the study guide. I think really the big key points here are the processes of mitosis and meiosis. I encourage you to compare and contrast those two with each other, and also reason out why you end up with these differences. So for example, you end up with 4 cells in meiosis because there's 2 rounds of cell division. You end up with 50% of the cells DNA in the daughter cells in meiosis because there's no DNA replication between these 2 rounds of cell division. You also want to know that mitosis, it was all about all other cells with the exception of the sex cells, and all about growth, repair, maintenance, things like that. Whereas meiosis was simply the production of the sperm and egg cell. Nowhere else do we see meiosis. You want to know our basic terms. We've covered them nicely. We discussed about the chromosome structure, and we got into discussing a little bit about alleles that are located at the same locus on the chromosomes. And the example of the different alleles that we had the widow's peak as an example. And we talked about the dominant and recessive alleles. So that's something to know. I will ask in the exam about the homozygous and heterozygous alleles, so you know homozygous, two of the same, heterozygous, different ones. In terms of the cell cycle, I think that it's likely that you will get questions about that. You remember most of the time in the interphase, mitosis was a short period of that, but an important period. Cytokinesis didn't include the division of the DNA, it included the division of the cytoplasm and organelles. We discussed about the checkpoints, and we also had a look of some of the control signals, control mechanisms of the cell cycle. We looked at karyotype very briefly, how male and female are told apart, and then we reviewed step by step the processes of mitosis and step by step the process of meiosis. And with the meiosis, we remembered that the big thing there was that we also have the processes of synapses and crossing over taking place. And we also finally compared and contrasted the mitosis and meiosis between each other. So that's all I have to offer in terms of the review for the exam 3. Remember, this is the last test that you have to complete. Please get it done in time so we can finalize your grades. And thank you for your attention on this video.